Hello, everyone. This is Chuan Jiao. I'm a PhD student in Paris. I'm going to introduce my project of demethylation in 2211 deletion syndrome and risk to develop schizophrenia. We have known that the 2211 deletion is a deletion on the long arm of the chromosome 22. 85% of the individuals have the 3 megabase deletion, influence about 60 genes. With this deletion, many organs have been affected. For brain, 40% of the individuals have deletion may develop to schizophrenia, to psychiatric disorder, and 25% may develop to schizophrenia. And schizophrenia as a multifactorial disorder affected by many genetic risk factors. This figure summarizes these risk factors and their effect size. 2211 deletion is here, and around it, we see there are also some other variants could have an effect on schizophrenia. Schizophrenia is a result of interaction between genes and environment. In general, this genetic effects on phenotype is stable during the lifetime, and the phenotype can be increased after the interaction with the environment by epigenetics changes. And this figure shows the gene-environmental interaction model of schizophrenia. Unlike most of the other symptoms of 2211 deletion syndrome, it needs long time to develop to schizophrenia. And the genetic changes in the very beginning, and then some environment factors like stress increase the effects to become schizophrenia. And the epigenetics was influenced by the gene-environmental interaction, which is a modification to DNA sequencing. It is variable during life and is heritable to the next generation, and it can change the phenotype by changing the gene expression. So let's say our genome is just like a book. The genetic changes can be a table, which can influence the meaning or influence our phenotype. And the epigenetic changes can be ink drop. It is changeable and could decrease the gene expression and then influence the phenotype. And sometimes in a short region, the methylation happens several times in different probes. We call it methylation region. So our aim is to find some biomarkers that show methylation differences in schizophrenia and non-schizophrenia individuals. First step, we check the demethylation differences between the 2211 deletion syndrome without schizophrenia and with schizophrenia using their blood. If you look at the differentially methylated probes between carriers with schizophrenia and without schizophrenia, there are many genes are affected, shown by the colored points. Some are higher methylated in schizophrenia and some are lower methylated in schizophrenia. We also found that some of them are already associated with schizophrenia in genetics and in expression and in molecular pathways. The same thing, but for the methylation regions, we found many genes. And here I highlighted the FGFR1 gene. In schizophrenia patients, the methylation goes down and the expression goes up. And this gene has been associated with genetic changes of schizophrenia patients and involved in the pathway of 2211 deletion syndrome. And second part, to control several factors like genetics, environmental factors, and age, sex, we have a chance to further study these monozygotic twins. The monozygotic twins are genetically identical, but for this twin carrier of 2211 deletion, one can develop to schizophrenia and another not, which we call the discordant monozygotic twins. It highlights the importance of epigenetics. So we did the correlation to see how exactly concordant they are in terms of gene expression and demethylation. As expected, they have a high correlation, but with similar genome and environment, there are still some genes are differentially expressed and methylated between them. And these genes are what we would like to know. And the third part, because we were studying the brain disorders, we would like to know what happened in the brain. So we took advantage from mouse model. We can get the brain tissue easily. We can have exactly same genome. We can control the environmental exposures. Like in this study, we put the mouse in a very high platform to make it in a stress situation. We have the mouse model with the with a region of deletion, which is similar to the human's 20q11 deletion. So to study the effects of the genetics and the environment, we have 4 groups of mice with deletion, without deletion, and without deletion stress, and with deletion stressed. We took their prefrontal cortex and extract DNAs, and then test their methylation levels, and to check their methylation differences between groups. So we got a list of genes for each comparison in terms of the effects of stress, deletion, and also their combined effects. And some genes are shared with genes from other comparisons. Although there are many genes, they contribute to the same biological functions, like the neuroprojection, morphogenesis, the dendrocyte development, the cell junction organization, and axon dendrite transport. At last, we combine all the genes from carriers with schizophrenia versus carriers without schizophrenia, and the twins and the mice. Totally 42 genes were overlapped, and they were uniquely highly expressed in brain. Means these genes mainly play roles in brain, and also for the specific brain cell types, they are highly expressed in astrocyte, which is a kind of cell type supporting neurons. And the most significant pathway of these genes is negative regulation of nervous system development. Means the genes we detect take part in the inhibition of the neuron system development. So in summary, 20q11 deletion with schizophrenia is the result of an interaction of genes and environment, which means even you have a deletion, there are still something to do, which can go in a good way if you have a support or a therapy. And the trajectory takes a long time. It can be changed anywhere by changing the gene expression and methylation for neural system, brain development, and cell junction. And also, we will try to do it in the future to study it. Our limitation is the sample size. Extending our cohort is necessary. For that, we need your help. If you have any clinical question, please contact this email. And if you have any research question, please contact another one. And thank you for those people who participated in our study and the researchers, psychiatrists in this work.